Hello again, class. Today we will be speaking about the polymerase chain reaction. The polymerase chain reaction is a biochemical method that is used to amplify DNA so that it can be analyzed by other methods later. In order to successfully complete the polymerase chain reaction, several reagents are required. First, we would need our template DNA or DNA that we are interested in amplifying. Secondly, we would need our primers of interest. Primers are short DNA sequences that are specific to the areas that we would like to amplify by the process. There are both forward and reverse primers that correspond to each complementary strand of the double-stranded DNA helix. We would require our polymerase chain reaction buffer. This is required to maintain the proper isotonic balance and pH that is required for PCR. We would also need our polymerase enzyme, which is required for the amplification of our DNA. We would also need magnesium chloride, which acts as a cofactor for the polymerase enzyme. And we would also need nuclease-free water, which would act to provide an aqueous environment for the PCR reaction to occur. In addition to this, we would also need dNTPs, which are the building blocks of our DNA, which would then be used by the polymerase enzyme to be able to build up our complementary strands and amplify our region of interest that can subsequently be analyzed. Shown here are the reagents that are required for our polymerase chain reaction. Our materials would include our micropipettes and tips and our very small PCR tubes. PCR tubes range in volume from anywhere between 0.1 to 0.5 milliliters. This one is a 0.2 milliliter PCR tube. Today, our objective is to combine all of the reagents and subject them to thermal cycling or cycling of various temperatures to be able to conduct the various stages of the PCR reaction. The first reagent that we are going to add is our PCR buffer. This particular PCR buffer is stained green so that we would be able to track the movements of our polymerase chain reaction reagents in subsequent reactions. We will add approximately 10 microliters of this reagent to our reaction. The next reagent that we will add would be our magnesium chloride, which acts as a cofactor to our polymerase reagent. This will be added at a volume of anywhere between 5 to 8 microliters per reaction. We will also add our dNTPs, which will be added at a rate of 1 to 2 microliters per reaction. We will then add our diluted DNA primers. Our DNA primers come lyophilized and must be reconstituted and diluted to 10 micromolar to proceed with our PCR reaction. We will then add anywhere between 0.5 to 2 microliters of each of our forward and reverse primer to our reaction. Our polymerase reagent or polymerase enzyme is always kept cold and thus must be added last, because while we are adding our other reagents, we would normally keep this reagent in the -20 freezer. We would normally add anywhere between 0.5 to 1 microliter of this reagent per reaction. We would then top up the remainder of the reaction with nuclease-free water to 50 microliters for the final volume of our reaction. After we have made up our master mix in our PCR tube, we will then place our PCR tube into a slot in our thermal cycler machine. Our thermal cycler machine is necessary for the completion of the polymerase chain reaction because it cycles the different temperatures that are required for various activities that are necessary for the completion of the reaction. The first temperature that is required is 95 degrees Celsius. This is required in order to denature our DNA and change it from being double-stranded into being single-stranded. The next temperature can range between anywhere from 50 to 60 degrees Celsius and is required for the annealing of the primers. Remember, our primers are our short pieces of DNA that are specific to our sequences of interest and also recruit our polymerase enzyme. The extension procedure begins at anywhere between 70 and 72 degrees Celsius, and this is where our polymerase enzyme will then be recruited and begin to recruit dNTPs to be able to extend our region of interest to make complementary DNA strands, leading to the subsequent increase in concentration of DNA that can be later analyzed. The PCR reaction normally cycles these temperatures over and over and over again until it has reached a satisfactory concentration of DNA. Most of the time, the PCR reaction has anywhere between 30 to 45 cycles of these temperatures. The various cycles, temperatures, and volumes of reagents used would be dependent upon the sequence of interest that is being interrogated and the source of this sequence. We can program our thermal cycler using our back, forward, up, down keys and our enter keys, and by reading the user interface to be able to either select a pre-prepared program for our PCR thermal cycler or to be able to develop a program of our own. After we have selected the program of interest, we then close our lid and begin our cycling process by pressing the enter button. A typical polymerase chain reaction will take anywhere from 1.5 to 3.5 hours. After our DNA of interest has been amplified by this thermal cycling process, it can then subsequently be analyzed by various methods. The method of analysis that we will be speaking about today is the gel electrophoresis method.